Alright, so let's start with a quick review of FSHD genetics, FSHD epigenetics, and then we'll get into some of the genetic testing. You know, the original Southern blot test, and then the more current technology that's used in clinically approved genetic testing. And we'll finish up with, a brief overview of the research testing that is epigenetic testing that we do through MyFSHD and here at the Jones lab at UNR. Okay, so let's get going. Okay, so let's start with some FSHD basics. All forms of FSHD are caused by genetic mutations that produce an epigenetic effect, and this is why I consider this ultimately an epigenetic disease. Okay, this epigenetic dysregulation leads to loss of repression and thus the gain of expression of the pathogenic DUX4 gene that's located on chromosome 4q35. So the key region of the genome for FSHD is the chromosome 4q35 D4Z4 repeat array. Okay, so you'll remember you have 23 pairs of chromosomes, so 46 total chromosomes, and you have one each from mom, one each from dad, so you have two chromosome 4s. And each chromosome 4 on the long end, the Q arm of the chromosome, at the very tip, there's what we call a D4Z4 repeat array. So a D4Z4 repeat unit, it's repeats of repeats. It's a highly repetitive sequence of DNA that is roughly 3,303 base pairs long. And this repeat is then repeated, head to tail fashion many times into what we call a repeat array. And this can be upwards of 120 or more repeat on chromosome 4. If you have less than 10, it's FSHD. Typically in the population, though, it's going to be 26 to 40 repeats or so. And since you have two different chromosome 4s, your two D4Z4 repeat arrays are most likely going to be different sizes. All right, so you have different alleles of your repeat array. All right. So FSHD is linked to this. It's very important, this repeat array. Now, there are other D4Z4s in the genome. And so here, these light blue, chromosome 10. Chromosome 10 is an almost near duplication of chromosome 4 repeat array. This is at 10q26. Now this D4Z4 on chromosome 10 is 98% identical to chromosome 4. Okay, and then there's a distal region that we'll talk about in a second. But the key is that FSHD does not link to chromosome 10. Most of the time, and there's a caveat to that I'll talk about. There's also other D4Z4s present in the genome at the acrocentric chromosomes, and they're just kind of scattered throughout the genome. And these are maybe 70 or 80% identical. Now the key is, if you're thinking about doing FSHD diagnostics or even just FSHD sequence-based research, you have to make sure that you're studying the right repeat, you're measuring the right repeat, and you're looking at the right repeat, because FSHD is linked to chromosome 4, not 10, and not to these other ones. So it can get somewhat complicated. All right, so I mentioned there's a distal region. Okay, there is. So distal to the array is a subtelomeric sequence, and it comes in two main variations, and one's arbitrarily called A, and the other is called B. Okay, and FSHD is exclusively linked to 4QA. Now we also have something we're going to talk about is 4AL, but oddly enough, the AL part, standing for long, is going to be here. It's not actually the subtelomeric part. Okay, so there's also a distal region on chromosome 10, 10QA, and 10 can also exist in an A or B. 10QA is not linked to FSHD. Okay, so now you have to make sure that you are now thinking diagnostically. Okay, what we want to know about is the chromosome 4q35 D4Z4 array in the context in cis, meaning on the same DNA sequence linked to a 4A or 4AL subtelomere. All right, so why is this? Well, FSHD is caused by a pathogenic activation of expression of the DUX4 gene, and, well, the DUX4 gene is present within the 4q35 D4Z4 array. Okay, each repeat actually has a portion of the DUX4 gene, all right? And DUX4 encodes a cytotoxic protein. Not exactly sure how it ultimately leads to FSHD, but we know expression of DUX4 is bad in skeletal muscle. But the key is, at least part of the DUX4 gene is in each one of these arrays. The second part is going to be distal here, and we'll be talking about that in a second because that's really key. All right, so only contractions on 4A or 4AL and not 4B lead to FSHD. Okay, so I mentioned that these D4Z4 repeats can be present upwards of 120 repeats per chromosome 4, normally again in the mid-20s to 30s. A deletion in these number of repeats to between 1 and 10, okay, in the context of a 4A subtelomere, that is by definition FSHD1. Okay. Now the same deletion in the context of a 4QB, that is not FSHD, and on chromosome 10, again, not FSHD. We'll get to that because why that is. Actually, this has been a really key event. Now the other part about that is, you notice it says 1 to 10. If you have no D4Z4 repeats, you also can't get FSHD. And why is that? Well, the DUX4 gene, that is the pathogenic gene in FSHD, is located within the repeat. So, you need part 2, the subtelomere. You also need part 1. All right, so you need to have this combination in order to have FSHD. Okay, so now this is the key paper in 2010. It came out of Sylvain Vandermeer's lab. Key author was Richard Lemmers, who is the world's leading expert, in my opinion, on FSHD genetics. All right, this is great work. Stephen Tapscott, Rabbi Tawil, Reno France, the gang Sabrina Ciccone, George Padberg, Dan Miller. Anyway, you see the names. This is actually star-studded paper and really significant because this is what nailed down the answer to the controversy in FSHD as to what gene causes FSHD. People couldn't agree prior to this. Now, some will say that, we always knew. Well, maybe you did, maybe you didn't, but suspected. But this is key because what this showed is, it had been known for some years from work out of, again, from Richard Lemmers and Sylvain Vandermeer, that contractions on 4B never led to FSHD, but only contractions on a 4A. So why is this? Well, it turns out that there's the sequence that's key in 4A encodes a polyadenylation signal, all right, for the DUX4 gene, and this signal is not present on 4B. It's also not present on chromosome 10. And so what goes on is when you have the contraction and the epigenetic dysregulation, the DUX4 gene is able to be turned on. The RNA polymerase transcribes the DUX4 gene, makes the mRNA that is spliced and processed. And then you need this polyadenylation signal, this little sequence. It's a 5-base sequence that tells the transcription machinery to basically stop and add on this string of 15 to 20 A's, polyadenylation. And this is important because this stabilizes the mRNA and allows the mRNA then to be transported to the cytoplasm and bound up by the ribosomes and be translated into DUX4 protein, right? And then you end up getting FSHD pathology. In non-permissive subtelomeres, they don't have this sequence. So the DUX4 gene can be activated, transcribed, spliced, but you don't get this polyadenylation. So the mRNA is unstable, degraded, no DUX4 protein, no FSHD. All right, so this, and on chromosome 10, which also is not associated with FSHD, it's a single base, and that single base change on the 10A subtelomere is right in the polyadenylation signal. You don't get polyadenylation. So this was actually spectacular work. It not only explained why the contraction only on 4QA or AL, which are pathogenic and not on B, but it also really nailed down that it is the expression of the DUX4 full-length processed and polyadenylated mRNA that is what's absolutely key for FSHD. Okay, so I keep mentioning 4A and 4AL. Okay, you'll see it written like this: 4QA and 4AL. So the subtelomere is actually the same. It's this last little partial repeat unit that exists. Some people call this 4A short, and this is 4A long. But you see this little difference here. Right? We're just talking about this is the most common in the human population, and this is then next. It's going to be this little bit longer. Why does this matter? It adds a little bit more length for diagnostics, which is important. For sequence-based diagnostics, it's really important because you need to make sure that that you understand the sequence you're assessing. And it's also actually important for gene regulation studies because if you're studying DUX4 mRNA, it does have a slightly different mRNA that it makes. And people have been misled by not understanding the sequence differences with 4AL versus 4A. But the key is they both are permissive. They both have the polyadenylation site. Okay, so that's actually what's key. Now, this is what throws everybody off. Okay, you keep hearing me talk about permissive. All forms of FSHD require 4q D4Z4 linked to permissive. What I mean by permissive is, 75% of the world population has a 4A or 4AL, at least one. 50% have two, right? And so why would that be? Because, 25% have two, 50% have one, and 25% have none. But you don't have FSHD. So it is not pathogenic. Having 4QA or AL, having this polyadenylation site, does not cause FSHD or lead to FSHD. You need other events to happen, but it's required for FSHD. So we consider this to be permissive for FSHD, where 4QB is non-permissive, because regardless of the other events that might happen in the region, if you're 4QB and you don't have the PAS, you're non-permissive, you can't develop FSHD. All right. So only contractions on chromosome 4 or 4AL or 4A 1 to 10, that's going to be FSHD1. And then also what's key is going to be that it really doesn't matter what your other allele is. And this is important because this shows you that FSHD is a dominant disease. One contraction on one chromosome 4 with a 4QA, that is actually causal for FSHD1, right? Doesn't matter whether your second chromosome's A or B, wouldn't matter. But then also FSHD2, which has other requirements, also requires 4QA or AL, at least one of the two chromosome 4s. It also has a specific requirement for size of a repeat array. We're not going to get into that. But the other key in FSHD2 is you need a second mutation on chromosome 18 in the SMCHD1 gene, which is an epigenetic regulator of the region. All right? And that'll be important later. This also explains why you need DUX4, right? FSHD2, which has somewhat different genetic mechanism to get to the epigenetic dysregulation, still requires an FSHD permissive subtelomere. So again, it's key. Some of the permissive 4QA or 4QAL is key for FSHD. And it's going to be key for diagnostics. And I'm just sick of seeing these diagnostic reports that people send me that don't check to see whether you're a 4QA or 4QB, because there are people out there that have a contracted 4QB, and guess what? You don't have FSHD. All right, because if you have two 4QBs, you cannot be FSHD1 or FSHD2. All right, a contracted B does not lead to FSHD, and a contracted B in the context of an A on a healthy non-FSHD chromosome is also not FSHD, at least not FSHD1. All right, so in FSHD2, BB, you also cannot have FSHD2. However, if you do have an SMCHD1 mutation that could cause FSHD2, you can be a carrier of FSHD2. So while you can't pass on the chromosome 4 genetics to your offspring, your kids, you could pass on an FSHD2 mutation. So if your parents have FSHD2, one of your parents, you could be a carrier and pass FSHD on to your children, even if you're seeming otherwise healthy. So it's important to get this checked out as well. And actually the epigenetic testing we do checks this out. All right, and why is this again? Again, permissive. All right, DUX4 on both of your chromosome 4s in the context of a 4QB, they're transcribed, they can be spliced, cannot be polyadenylated, unstable, degraded, no DUX4 protein, no FSHD, both chromosome 4s. Doesn't matter if it's FSHD1 or FSHD2 that you're thinking about, you can't have either one, or even FSHD3 that's coming down the path. All right, so your FSHD chromosomes, FSHD1 is going to be linked. All right, chromosome 4. Now, I told you, I said it's exclusive to chromosome 4. I was very careful. Exclusive to chromosome 4QA or AL D4Z4 repeats, because there is actually a chromosome 10 FSHD1, but it's only if this is a hybrid chromosome that has a chromosome 4 D4Z4 distal unit and a chromosome 4 subtelomere. All right, so how does this happen? Well, it turns out, I mentioned that the D4Z4 and chromosome 4 and chromosome 10 are 98% identical. All right, well, sequences, especially repetitive sequences at certain points during development, very early in development, whether sometimes it's spermatogenesis and oogenesis, and even at 1 and 2 cell stages, you can have a little bit of recombination, rare event. But what happens is you get a recombination between the very similar sequences, and this leads to a hybrid chromosome. If you had a contracted 10, which is not pathogenic, and a healthy 4, they recombine, you end up with a contracted 10 that has chromosome 4, D4Z4, at the distal tip, and the permissive 4QA or AL. It could happen with AL as well. And that actually is FSHD1. All right. And the key is here is this is often misdiagnosed by southern blotting and optical genome mapping. There's up to like 5% of FSHD1 cases are this hybrid chromosome. All right. Now, sometimes it does get picked up. It all depends on how many repeats. You know, this could be 5 chromosome 4 repeats and 1 chromosome. You know, it matters. But it can be missed. So if someone is clinically FSHD, and this happens actually, this does actually happen where, you know, really good FSHD clinicians, good, really fantastic neurologists that really understand FSHD will come to us and say, you know what, this person, I know they have FSHD, but the genetic test came back negative. Can you take a look? And we'll do our epigenetic test, and you'll see in the end that we're actually able to pick this up. And I can say, you know what, I bet they have a contraction on a chromosome 10. Yep, they got a contraction on chromosome 10. You know what? It's a chromosome 10-4 hybrid. The epigenetic diagnostics can actually pick that up. The key is we just want to help people get it right, but this is something that's got to be careful for. All right? So FSHD2, this is going to look otherwise like a not FSHD chromosome. Yeah, okay, 4QA, but you have between 8 and 20 repeat units. The key is you need this a pathogenic mutation in the SMCHD1 gene. Now, there's probably 20 different genes that can lead to FSHD2. More than 80% of the time, they're in SMCHD1. Very rarely, they're in other regulators, DNA methyltransferase 3B, LRIF1, and actually we haven't found the others. But most often it's SMCHD1. Regardless, the effect is going to be an epigenetic effect here that we're able to pick up with our epigenetic diagnostics. All right, so then there's the not FSHD chromosomes, greater than 10 repeats, doesn't matter whether it's a 4A or 4B, contracted 4B, contracted 10, and of course all the other D4, Z4. Now, you know, I throw these in here because, you know, the sequences are similar enough that as people are doing sequence-based diagnostics, there's actually a proposed diagnostic out there that picks these up and actually gets it wrong, because the thing is, these D4Z4 repeats are not epigenetically dysregulated in FSHD. These actually are epigenetically dysregulated, all right? So if you're looking at epigenetics or any sequence-based diagnostics, you got to make sure that you're not studying just the garbage repeats that aren't associated with FSHD at all. Just got to be careful. That's actually a key with FSHD. The devil is in the details on everything FSHD is. A complicated disease. Gotta pay attention. All right, so what about diagnostics? Well, you know, mention that the definition of FSHD1 is 1 to 10 repeat units. Well, the original diagnostics, and this was all figured out in the Netherlands, and again, the Netherlands is kind of the home of FSHD research and diagnostics and almost everything FSHD. Some people might disagree with me, but I think pretty safe to say that. What there was a physical measurement of the size of the D4Z4 repeat. That's traditional FSHD genetics. Just we know that this is 3,303 bases, and we know that 1 to 10 leads to FSHD. So let's just measure the DNA. All right. Now remember, you have the chromosome 10, D4Z4. And, you know, you're going to be, so you're going to have to distinguish, you know, going to have chromosome 4 and chromosome 10. You have 2 of each, so you're going to have 4 sets of D4Z4 repeat arrays to study. You're going to be looking for a short one on 4, only with A, right? And FSHD2, well, measuring it's not going to help you a whole lot with FSHD2, all right? So the key is going to be that there are these things called restriction enzymes, and one of the important ones we're going to use is called EcoR1. These are isolated from bacteria. It's kind of a genome defense system in bacteria. So E. coli strain R, enzyme 1, will digest DNA of the sequence GAATTC. So remember, there's 4 bases of DNA, GATC. And so 4 to the 6th, it's a 6-base cutter. Every 4,096 bases on average in a genome, you should have an EcoR1 site. Now that's going to be skewed because this is a repeat. So you isolate this from the bacteria, And we use this, and so you're going to isolate blood from a patient, and then get the genomic DNA. You need very good high molecular weight DNA. DNA is actually pretty fragile, and will break. You want to get this whole region intact. And here there's an echo R1 site in the 4A subtelomere, and there's an echo R1 site flanking this on the centromeric side. And we know that this is how much DNA there is here. It's about 5,000 bases and then a few thousand down here. And so we know that echo to echo is about the greater than, say, 40 kb is going to be more than 10 repeat units, and that's healthy. All right. Now, the thing is, you have that exact same setup here on chromosome 10. All right. And then you're going to see it's going to be much smaller when FSHD1. And again, FSHD2 is going to be kind of hard. So you're going to cut with echo. I call it, some people will say ECOR1. I call it echo R1. We're going to cut it with echo R1. You're going to get this, and here you got, so all these types of repeats. So how do you figure that out? Well, it turns out there is another restriction site that's going to be in there that's going to be very useful, and that's going to be one called BLN1. You see this little B here. Okay. Now the B, I told you that the chromosome 10 and chromosome 4 are 98% identical. Well, it turns out one of those spots where they're not identical actually is a different restriction site. And you can see that that's going to be cutting here, and we're going to be able to distinguish chromosome 10 from chromosome 4. All right, so we're going to cut with echo R1. First, you just do echo R1 by itself. That's going to give you all 4 arrays. Then you're going to cut with echo R1 BLN1. All right, and so that's going to cut here at the B. It's going to cut here. It's going to cut here. So all 4 are going to get shortened a little bit. And then the chromosome 10 ones are going to get totally chewed up. They're going to get a cut every place. And that's going to, not totally chewed up, but they're all going to be about 3,000 bases. And this is going to drop, be much smaller in size and somewhat disappear, because your tag is actually, the tag that you're going to be probing for is just right here. So basically this is going to disappear. So what you'll be left with are 2 chromosome 4 arrays. All right. So what does this look like? Well, this is where we're going to use a technology called the Southern blot. All right. Now, you're a little confused. The Southern is was Dr. Southern at Edinburgh University. I think it's Edwin. I think his name was Dr. Edwin Southern. So that's why it's called the Southern blot. In 1973, if I'm correct, developed this technology, I believe, mostly for paternity testing. Which how do you track chromosomes? And so it's got nothing to do with FSHD, but the Southern blot technique is kind of depicted down here. What that is, the thing you have to remember is this cartoon looks really easy, right? You have to find your chromosome 4 and your chromosome 10s. Well, remember, there's 3 billion bases of base pairs of DNA, on the haploid genome, so 6 billion bases of DNA, that are also getting cut by echo R1 or echo and BLN1, right? And you have to somehow be able to find the specific sequence you want in the background of the rest of the human genome. All the other chromosomes all cut up, and they're all in the same tube together. So how are you going to do that? Well, the way you're going to do it is cut with your restriction enzymes, and then you're going to separate the DNA fragments by electrophoresis. So DNA is negatively charged, so it's going to run towards the positive, and you run through an agarose gel. Agarose is isolated from kelp, actually. I guess you can eat it even. It's kind of like gel. And based on the percent that you run, you're going to separate the DNA fragments based on size. And the smaller ones move through the gel much faster, and the big ones get held up. And so you end up, now you're separating all the DNA based on size. Okay, you're also going to run a marker that tells you exactly the size. So now you have a setup. You have all your DNAs that are cut with echo R1 or cut with echo R1 and BLN1, separated by size, but you still can't find them. Right? Well, then the next thing you're going to do is you're going to need to somehow have a probe. And figure out how to find it. So you're going to, the key to the Southern blot is you're going to denature this DNA, okay, with sodium hydroxide. Double strand DNA becomes single strand. And high salt is going to be stable as single strand. You're then going to transfer this to a nylon or a nitrocellulose membrane, essentially like a piece of paper. That's all the DNA is going to come out of the gel onto a piece of paper and stick there and stay there. All right, but single stranded, which means G can bind to C and T can bind to A. And so if you know the sequence you want to look for, you then take a radioactive probe and you're going to label it, make it single strand, and it will find the sequence that specifically you're looking for and make it radioactive, expose it to film, and you get an autoradiogram that it will show you the sizes of the DNAs that you're looking for. All right, and that's basically what Southern blotting is going to be. All right, so let's go through it. All right, you're going to cut with Echo R1. You get your four things here. All right, now the key is we got a probe. Okay, this is P13E11. This was actually the key. You had to find something that was unique in the genome that's only going to pick up these 4 arrays, and that's what this probe does, P13E11. You radio label it, okay? And so you have your Echo R1 fragments radio labeled. You go, now when you do the Southern blotting, you're going to get something here that's maybe 140 kb, kb being 1,000 bases, so 140,000. This one looks like it might be 110,000. Maybe this one's 60,000. And this one here looks like maybe it's 35,000. Okay, so you got a short one. Now you don't know if this is chromosome 4 or chromosome 10, but you got a short one, and that's interesting. So now there's the potential that you have an FSHD1 situation. All right, so now you're going to go with Echo R1 BLN1. You're going to cut with Echo R1, you're going to cut with BLN1, and you're going to now go through and you're going to look. And what's going to happen is you do your P13E11 probe. Now, the chromosome 10s are running off the gel. They're actually not even on here, right? And you're only left with the chromosome 4 Echo R1 BLN1 fragments, right? Chromosome 4, chromosome 4. So this short one, was actually chromosome 4. So now you know you have a short chromosome 4. Okay, so that's good news. Now, what you don't know at this point is if it's 4A or 4B, because guess what? B also has a little element, an echo site down here too. And so you have to do another Southern blot. You should do another Southern blot if you're a good lab. Now, sometimes they don't, but you come back and you do a second probing for 4A or 4B, and you can determine whether or not it's the short one's an A or B. Oftentimes that's skipped, unfortunately, and it can lead to some misdiagnosis. All right, so there you go. That's what's going on. So what about this situation where it's chromosome 10? Cut with Echo. All right, the situation here, Echo, there's your 4 sites. There's your short one. You don't know if this is 4 or 10. You cut with Echo and BLN1. Only chromosome 4 is left. The 2 big ones are chromosome 4. This short one was a chromosome 10, and so that is not FSHD unless it has that distal element that maybe is a recombination that would have been missed on this test. Okay, because that recombination, if it's missed, think about it, the probe is here, right? The probe is going to be up here, and it only goes to the first cut. So unless the recombination was a complete recombination, that probe is not going to pick up, even if the fragments are not digested in that chromosome, in that hybrid. All right, so that's where the misdiagnosis comes in with the Southern blotting. All right, now there's newer techniques that are out there. They have the same sort of needs, meaning you need high quality, high molecular weight genomic DNA, because these new techniques are going to do the same thing as a Southern blot. It's kind of like automated though. They're going to measure the size of the D4Z4, and they're going to detect whether you're a 4B or a 4A. They're going to use fluorescence. All right, so molecular combing, and I think the company may not be doing so hot. The only place I knew they were doing this was France, and I think they stopped doing it. But regardless, just so you're aware, if you've had this before, so green is the a probe that interacts with the D4Z4, hybridizes with D4Z4, and so you can measure how much green you have, and that tells you how long the D4Z4 is, and then you have specific colors that tell you whether you're 4A, 4B, 10A. And from what I understand, when this works, it works really well, but it didn't work very well very often. Anyways, my understanding, don't get mad at me if you're with the company, technology is kind of cool. But more recently, there's single molecule optical mapping, and BioNano has put this out, and this is seems to be what's kind of going more common in diagnostic labs now around the world and in the U.S. And so it's somewhat similar. You actually are going to nick the DNA and put fluorescent probes in, and you're still going to measure the 4QA, D4Z4AL. You're going to measure chromosome 10, 4B, 410, 10A. So you can measure, and you get a lot of information here too. Find recombination genome. Now, this can still and does still miss the 410 recombination, or the 10 chromosome 10-4 recombination that can be pathogenic, and I still get some of these. But in general, this is the same idea. You're measuring the size of the D4Z4. You need high quality, high molecular weight DNA from blood, fairly automated. The Southern blotting takes a lot of skill. Leiden University Medical Center is probably the best in the world. University of Iowa Diagnostic Lab is really fantastic as well. But I believe they're also switching to BioNano. And then the last one that's coming along is this thing called long-range sequencing, put out by Oxford Nanopore Technologies, where you literally sequence through the entire region, and you start with a 4A, and you sequence through the whole D4Z4 and come out the other side, and you're able to tell how many repeats are there, any mutations. You also get some methylation, some epigenetic stats. Again, though, you need high molecular weight genomic DNA from blood. This is not as straightforward as it looks in this cartoon. There are some technical aspects to it that are a little tricky, but it does work. And it doesn't work in all sizes, but anything under 1 to 10 repeat units you'll be able to get. You need a little trouble maybe some of the larger FSHD 2-1. Although I've seen data up to 24 repeats, so it's possible. And now the technology for the sequencer is actually coming down in price. Now, the key is these top two, so the three of these technologies don't change the game from Southern blotting in the sense that you still need high molecular weight blood DNA that's properly processed and very specialized equipment. All right, the price of these technologies has not come down, so you haven't increased accessibility, and you're not able to do it on saliva DNA. Now, the price of the nanopore sequencing may be coming down because they have different little portable sequencers and stuff. We'll see, but that's not quite being used as a diagnostic yet. All right, now what we do is something a bit different. So all forms of FSHD are caused by genetic mutations. We focus on the epigenetic effect, right? All of those are looking at the genetic effect. We're looking at the epigenetic effect, all right? And so epigenetically, a genetically not FSHD chromosome 4 has a epigenetic status that is epigenetically repressed, transcriptionally silent, for the most part, and highly methylated on the DNA. And it doesn't matter whether it's a 4A or 4B, right? FSHD1, you have one chromosome 4 that is epigenetically unmethylated or low levels of methylation, and the other one is healthy levels, again, methylated and off. And in FSHD2, both chromosome 4 D4Z4 arrays are epigenetically dysregulated. The yellow means there's no methylation or very low methylation. The black means high methylation. So if you just think epigenetically, and you go through and you look, you have methylated, methylated, unmethylated, methylated, unmethylated, unmethylated. All right, and that's the basis of epigenetic diagnostics. It's this distinct FSHD epigenetic profiles. All right, so, right, I guess saying it again, epigenetic diagnostics is based on these epigenetic profiles. All right, so what are we going to do? Oddly enough, the first thing you got to do, the first thing we do is something that's a genetic analysis. First thing you want to know is, do you have an FSHD permissive 4A or 4AL? We don't care whether it's on or off, methylated or not. We want to know, do you have one? Because if you're BB, we immediately know it's not FSHD. And the second basis of that then is that our epigenetic assay is based on a permissive chromosome 4. We don't care about the epigenetics of a 4QB distal repeat unit. We look at the epigenetic status of the last D4Z4 repeat unit in the context of a 4A or a 4AL, because this is actually the repeat unit that leads to DUX4 expression and is the pathogenic repeat unit. All the other ones, who cares? This is the one that matters. All right, so we're going to say, what is the epigenetic status of 4A or 4AL, right? And then the next thing we're going to look at is, okay, if you're FSHD, is it FSHD 1 or 2? And we're going to look at the epigenetic status now of all the D4Z4 repeats in chromosome 4s, and you can see how very quickly you'd be able to pick it all up on whether it's FSHD, then it's 1, or FSHD 2 by how many methylated repeats there are present. In FSHD 1, most of the repeats are still methylated, because the small, think about this. If this is 50 repeats long, and this is 5 repeats long, less than 10% of the repeats are epigenetically dysregulated, right? So you're just going to be mostly methylated, where in FSHD2, they're all dysregulated. Okay, so that's the basis of epigenetic diagnostics, right? And the key is it can be performed on low-quality saliva DNA, because these are all small. We're not measuring the size of the array. All we need is still 500 base pairs pieces of DNA, low quality saliva DNA that can be collected through the mail anywhere in the world, stable for years. You can actually pull DNA out of the freezer. We've assayed DNAs from the 80s that have been in the freezer for 30 years. Is that right? 80s, how far? Yeah, I guess like 30 years. Before there was a diagnostic for FSHD, and we can actually test those for their methylation state. You can't do it with any of the other technologies because they're fractured. Defreezing DNA is really bad for genomic DNA in respect to keeping it the size and the integrity. All right. Now, one thing I want to bring up is we focus on epigenetic in the FSHD. There are genetic definitions of FSHD, right? So by definition, FSHD1 is 1 to 10 repeat units. 9 repeat units with a 4A, 10 repeat units with a 4A, 11 repeat units, and you're not FSHD. Now, even if you're clinically FSHD, you won't be considered genetically. You come back as a negative, all right? Now, we look at it slightly differently. I would look at it and say, if you're epigenetic, if you're clinically FSHD, I don't care whether you have 9 repeats, 10 repeats, or 11 repeats. These situations would come up positive by epigenetic testing. These would be negative by epigenetic testing. I actually believe that these are and should be considered positive. And so sometimes people get a result through the MyFSHD testing or through my lab's epigenetic research testing where we give them you're positive for FSHD, and they get a negative result. But then I find out that they have 11 repeat units or 12 repeat units. If you're clinically FSHD and you're epigenetically FSHD, I don't care what your genetics are. You should be FSHD. All right? And that's kind of what's going on here. We get this conflicting FSHD epigenetic and genetic results, and this causes some stress with clinicians and patients. Now, I don't have any patients because we're researchers and we just have subjects. And we only do a research test. But if someone is clinically FSHD, but they're not genetic FSHD1 because they have 11 to 14 repeat units, they're not genetic FSHD2 because they don't have an SMCHD1 mutation, they may have FSHD2 levels of methylation for various reasons, but no mutation, but they are positive for epigenetic FSHD1, situation like this, where one chromosome 4 is 11 repeats, 12 repeats, but epigenetically on. The other chromosome 4 is pretty close. I'm kind of epigenetically on, kind of off, maybe don't really know. If you're clinically FSHD, and this is the situation, I think it should still be considered FSHD. And the problem is these individuals, they're going to be searching for what is their disease. They're going to be doing other diagnostic testing. They're not going to be able to get into clinical trials. They're not going to be able to get therapies. And we got to fix this because I believe epigenetic FSHD and the combined with the 4A, 4AL is actually combined with clinical FSHD should be considered FSHD.
Okay, well, there's your FSHD1 crash course. I know it was a little long, 40 minutes or so, but you know what? FSHD is complicated for researchers, for patients, for your medical doctors as well. It's just a complicated disease. And I just want to make sure it's very clear, something you can go back to understand. Because understanding the genetics and the epigenetics, I think will also help you understand how this plays out in yourself or your children or your family. All right, so if you have any questions, as always, please feel free to send me an email, peterjones@med.unr.edu, and we'll set up a call or a Zoom. I'm happy to go over questions. You can try calling my office, but I gotta tell you, I'm in and out of the lab. I still work in the lab, and so it's really best. Send me an email, and we'll set up a time. And I'm happy to answer anything about FSHD. Well, anything that I can. I'll do my best. Again, it's complicated, but the field's moving fast, and you know what? We're gonna get there. We're gonna get there pretty soon. All right.